Hi, morning everyone. As Marcellus has said, that I'm working as a rust pathologist and molecular geneticist at CIMMYT. I'm based in Mexico headquarters. So today I'm just going to take you through a part of my journey that involves a lot of field challenges involving rust pathogens and also kind of like use of the genomic tools to deal with this. So this is kind of like giving you a glimpse of the work that I have started doing since I've joined in CIMMYT, and the work that I've been doing to address the wheat rust threat, and also using different tools to kind of like future-proof the elite wheat germplasm, which is very important across the globe from CIMMYT. So the work is based in, is like kind of like grounded in Mexico and Kenya, but the lessons go far beyond these countries. So I think all of us, most of us who are present here, we do know, we've been hearing about the talks from like about the rust. And what makes it more particularly formidable? We know that the diseases are very, very important, and in case of wheat, because there is a lot of area under production for wheat, so the yield is very, very important. And among those diseases, rust are the most devastating disease. Why? Because they have a history of traveling long distances, they can evolve rapidly, and within weeks or within days, if there is like the conducive environment present, they can just like knock down the crop within days and weeks. So there are like different estimates for the global annual losses for the epidemics that have been recorded for the rust diseases around the globe. But that also depends on like this susceptibility of the cultivar that was present there, the one that was the epidemic was caused on, the level of inoculum that was present. So it can cause huge yield losses, and then in the end there is like nothing that a farmer can take to the field. So stripe rust races that are more aggressive and that got adapted to the warmer climate have migrated and spread towards across geography since 2000. This is a map that was adapted from Sajid's paper, where he showed like the kind of like migration of the PstS pathotypes. But over the time, a lot of arrows have been added into it, and then race shift towards the higher level of mutations for virulences within these PstS populations have actually resulted in the vulnerability of the widely deployed cultivars around the globe. And if you look at the graph, like it shows like there are a lot of arrows going up and down, up and down. So a lot of these actually shows the exotic incursions that we have experienced in case of stripe rust. We have had exotic incursions where the PstS populations have been replaced within US since 2000. And also some of the European PstS pathotypes getting replaced by the races from the Himalayan region. And also we have a lot of history in Australia where there have been exotic incursions of stripe rust from Europe as well. So all this kind of like shows you the intercontinental migration of these races. And it shows that like how this results in the kind of like it puts the host resistance deployment at a very risky level as well. And it's not only like I've talked about US or Australia, Europe, but then if you remember Sylvia's talk yesterday, so South America, like in Uruguay, there was no epidemic or there was no stripe rust before 2017. But after Argentina experienced the epidemics, they also got the incursions of stripe rust in Uruguay as well. So all these kind of like things shows you that the migration is only a matter of time. So it is like, this is what history has shown us, that there is like the faster continental spread that's happening around the globe. And even within the countries, there are localized severe epidemics that are happening. And we have seen that every alternate years, you'll have like, one or more genes getting broken down. So I'm just going to talk about that PstS16. It is not like relevantly new race, if you remind yourself of the Dave's talk as well. It was first detected in Afghanistan, Pakistan in 2018, and then it was also detected in Ethiopia in 2020. And recently, we have had an outbreak of PstS16 in Kenya. In Kenya, it was the most predominant PST pathotypes that were present were PstS2 and PstS11. But in 23, we have seen the devastation the PstS16 caused in Kenya in our international phenotyping platform. And then we are hearing about that there has been this has been one of the most durable major gene, YR15, that recently got broken down in UK as well. And of course, you're going to hear more about that later as well. So all these events kind of shows you that the breakdown of a resistance gene is not an independent event. So it's not like a single event that will happen wherever it has happened and that's it. It doesn't work like that. It can move to countries, it can move to continents, and this is something that we need to be prepared for. So I would just like to show you that this picture was taken in 2022 in Njoro, Kenya, which is one of our international phenotyping platform, along with Calro. And so at that time, this is the CIMMYT material that was being planted there for testing. A lot of their stage 1 and stage 2 material actually goes every year for evaluation. So you can see that at that time, the predominant pathotypes were PstS2 and 11, and you can see that how the field looked like, the trials looked like. And then in 2000, this was supposed to be 23, sorry, not 24. So that was the main season, and only in a matter of 6 months, you could see that the field became all yellow. So most of the CIMMYT germplasm, I would say about 40 to 50% became susceptible to this race in Njoro, Kenya. These were just specific, some of the lines that became susceptible, and among them was the Borlaug 100, which was a very resistant cultivar, and it was released in a lot of countries. And it stayed resistant until 2022, but in 23, it got broken down. And even now, the recent data that I have collected, it has gone up to 40, 50% of the severity that is showing in the field in Njoro, Kenya. I'm just going to move to the stem rest. We have heard a lot about stem rest from Dave as well, from Diane as well. So it's just it's almost the similar things, but kind of showing that after UG99, we have had a lot more lineages that are developed, mutated from UG99 as well, but then we also have non-UG99 races. Although stem rust has always been a disease of more warmer, humid climates, but then also because of the higher altitude, higher rainfall areas have become the hotspot for this disease as well. Why? Because of the continuous cultivation of wheat that we see in East Africa, in the highlands, and then also the survival, the build of the inoculum around the year because of the presence of the host plant over there. So it is not only limited to East Africa, but it has moved to there have been a lot of reports of the epidemics in Europe as well, non-UG99 lineages, but still they have caused epidemics, taking over a lot of stem rust resistance genes. And then recently, as Dave mentioned as well, in Nepal, there was a report of UG99 in 23 as well. And then I didn't put the Bhutan as well, but they've also mentioned that too. And just to kind of show you that, this is one of the varieties that was released in Ethiopia with this 7.5 tons per hectare yield potential. And then when the race got mutated, it took out the SRTMP, which was the major gene in there, and the yield losses were 100%. It completely took over all the acreage that was under Robin. Now we use this as a susceptible check in our trials. Just wanted to show you a bit about like the CIMMYT breeding pipelines, that how CIMMYT has changed a bit of its, how they are actually targeting their target environment. So they have made it, so earlier I'm sure that a lot of you are familiar with the shuttle breeding that Norman Borlaug started. So shuttle breeding between Mexico, Obregon, Toluca, and Kenya, Njoro. So we have taken a step forward from there. So earlier in that shuttle breeding, Mexico would send the breeding material, the F2, F3 generations to the Njoro, Kenya for selections as well. But now we have split the pipelines to be more effective, more efficient. So we have like these 4 pipelines are based in Mexico, targeting different geographies, different agroecological zones. And then this pipeline 5 is the East African breeding pipeline that is based in Kenya. So actually, we have made East Africa as a hub for that breeding efforts, so that all the efforts that are being made are on the ground with all the new virulences and then all the new, we don't know that what's happening with the pathogen evolution. So it is better to just start from the scratch on the ground and go along with that, along with all the seasons, to see that, so that we can have the better lines, varieties to be distributed across Eastern Africa. But we also have, so this pipeline can also cater to some of the South Asian countries as well, because some of the virulences that we have in East Africa does kind of like go, are similar to, somewhat similar to the South Asian virulences as well, in striiformis majorly. So across all the breeding pipelines that we have, it depends like the different agroecological zones. But if you see, no matter which pipeline do you have, rust remains the most important trait for CIMMYT to be deployed into the lines that we are developing at CIMMYT. So all the elite germplasm that gets sent to different countries in South Asia, in East Africa, any country. So we need to have, the crosses shows the level of importance. So all the 3 rust, depending upon the agroecological zones that these breeding pipelines are targeting, are the most important. And even in, I know that like in India, in Pakistan, Bangladesh, and I'm pretty sure in East Africa as well, until and unless, even if the one or the other rust is not important, according to the climatic conditions, you still need to have a desirable level of resistance for that line to be released as a variety in that country. So that puts it kind of like at the upfront that this is the very important disease trait to have in the, your breeding material. Okay, so just to show a bit of I think the strength of the CIMMYT is its international collaborations and the international phenotyping platforms that we have across the globe for different diseases. For rust, we have a phenotyping platform for leaf rust in Obregon, Mexico, for striiformis in Toluca, Mexico, and then we also have phenotyping platforms for striiformis and stem rust in Ethiopia, also in Njoro, Kenya as well, and then also a bit of Uruguay as well for the Fusarium head blight, Septoria, leaf rust, and as you have heard about the blast that we have in Bolivia, we have a phenotyping platform, also in Bangladesh, in Zambia as well. So this is what provides the strength to the CIMMYT breeding breeding pipelines, that the material that gets sent to the countries is actually covering not just one trait, but the multiple traits that we are working on to cater to all the regions that we work for. So just wanted to put a little bit of that, although because we are covering different regions in the world, the virulences are different. There was a time when the Mexican pathotypes were the most virulent. That's not the case today. So we have good virulences in Mexico, but now I think the virulences that are there in East Africa and in South Asia have mutated much more. They have evolved much more rapidly than what we have in Mexico. One of the reasons for that is as well because the belt, the north of Mexico, where we grow majorly wheat, is like mostly the climate is conducive for leaf rust, And we have some local stem rust races. But then Toluca is the only place we use for the phenotyping. But it is like a hotspot for yellow rust, but there is no wheat grown around that area as well. So it doesn't get that conducive environment to actually mutate that rapidly as compared to the East Africa, where the wheat cultivation is around the year, or South Asia as well, where we have like a conducive environment for the stripe rust. So this is just like for PstS16, some of the parental candidates for the bred wheat from the breeding pipelines to show the level of resistance that we had. So some of these lines, so we have, so now from Mexico, we do get like the stage 1 lines, which are your parental candidates. So it's about like 5,000 to 6,000, depending upon like how many lines do they have. And also like we have about 1,500 to 2,000 for the stage 2 lines as well. So among the huge data set that year that we had, we did have some good level of resistance in the lines, although as I said that 40 to 50% was mostly like wiped out on the ground. And so now PstS16 is the most dominant in Kenya. Although if you look at the virulences, it is not a lot different than what you can see in PstS11 or PstS2. So our fear is that it is taking out a lot more than what we can actually see, or like depending upon the differential sets or the near isogenic lines that we have. So that is the kind of like thing we are worried about, that we don't know exactly, because if it's taking out a lot more of CIMMYT germplasm, and most of the CIMMYT germplasm has adult plant resistance genes as well. So the known genes, we can see that, okay, what on the basis of the pedigree, some of the marker information of the genes that we do put in the crosses, we can see, but then there is something else happening with the PstS16 as well, that it's taking over a lot of germplasm. And then stem rust races in Mexico compared to we have very, very localized stem rust. You don't see a lot. It's majorly leaf rust, but not a lot of stripe rust. But East Africa, yes, you can see like in Kenya, it is a hotspot for UG99. So out of the 17 races that have been evolved from the original UG99, 12 were actually detected in Kenya. And then 7 evolved in Kenya through mutation as well. And then in Ethiopia, 6 races were, have been reported and detected in Ethiopia. So this was just the same for the stem rust material, that we do have like a good level of resistance, but mostly the resistance is based on the major genes that we have in CIMMYT germplasm. So we do have these, the presence of 13, 22, 25, 26, SRND, 50. So combination, so it's never one gene, it's either 3 or more combination of genes that we have, along with some APR QTLs that are in the background that have not been fully characterized as well, but just on the basis of some of the QTL mapping results. So it is there. So for stem rust, it is majorly, even until now, based on the major genes. So just going to touch base a little bit. This is something that after we had PstS16, we actually made a panel of the lines that to see that the lines that actually survived PstS16, to try to figure out that what is there that is providing resistance when most of the material actually got knocked down because of that. So also, and also for the stem rust as well. So we had like a panel of about 829 elite bred wheat lines from our breeding program that we selected, and then all these lines are the ones that will eventually be sent to the partner countries around the world. So what we did was then we selected that panel and we phenotyped it for 2 seasons, in the off season and the main season in Njoro, Kenya. And then we genotyped using the 40K XT Infinium SNP chip, which is developed by the Agriculture Victoria in Australia in collaboration with Integrain. So it says 40,000, but the 40,000 SNPs are not only for wheat, they have a chip for the dual purposes. So about 25,000 is what they have for the wheat, and 20,000 is what they have for barley as well. So you can do both the crops on the same chip as well. So this was the kind of like frequency distribution. This data was something that I got recently, so it's a very preliminary result that I will be showing. So this was like what we found in the field for the stem rust and for the yellow rust. So you can see like some good level of resistance, which shows you that there are in the background some R genes, the major genes are present, and then also like some good intermediate level of resistance that shows the indication of the APRs that you have. Just the kind of like kinship matrix, all of you must be familiar, and then also just to show that because the blood that we have in CIMMYT breeding is like, you know, highly relevant. So we have like, you will find that the parentage pedigrees goes way behind. So that's why like they are very, very relevant, you can see, but still you can see some level of diversity in those lines that are coming up in the recent the breeding program. So for the stem rust, we got like a lot of hits. My initial filtering was a bit stringent, but still I got really good kind of like hits on the good marker trait associations as well. So the kind of thing that I had a look at it is try to put that what I'm getting, but this needs more refinement as well. But the good thing was that kind of like made me very confident about the results was presence of these genes, that some of these regions that I am getting, we do have that in our breeding lines based on we can see that based on the pedigrees. We can also some of those, because we also have a marker introgression pipeline that where we are introgressing these genes deliberately in our germplasm as well. So most of these genes were the MTS that I got were like kind of like confirmed that we do have these in our pipeline. Some of the others, not so much, because that's not something that we are deliberately putting, but maybe there can be some QTL that already have been identified and reported, so that could be also present depending upon the pedigrees that we are using. So this is something that needs to be kind of like looked into, and then we'll take it forward from there, that if we are finding something novel, and to take it from there. Okay, for stripe rust, it was not as much as what we found for the stem rust, but still it was a progress. So for stripe rust, we, as most of the people have mentioned, that we do have 2NS translocation. 2NS has YR17, LR37, SR38, but that is not effective in Kenya because we have a virulence for YR17 in East Africa. So that's why we didn't get any region for the 2AS, so which was understood as well. But then YR29 was there, and about 99% of the CIMMYT germplasm has LR46 YR29 fixed in it. So you will get it, although the level of the resistance varies, but still you can pick it up with depending upon the year that you are having when you did the testing. And then YR78 is another gene that CIMMYT has started putting into its background as well. So it was also kind of like indication, because like the marker that I got was like linked to the same region as well. So another one was 6A, YR81. It is present on the short arm right at the top, but this was identified and characterized in a Watkin landrace in Australia. So I'm not sure how true that is. This is something that I need to kind of like figure out if it is the same gene or if it's not the same gene, doing some marker testing and stuff like that. So we do have like some of the other regions that I did get. Some might be corresponding to the QTL that have already been reported, but some might be novel. And those are the ones that we are really interested in against the PstS16. So apart from that, another thing that I've started doing, I will always work on the discovery and characterization, making the biparental populations. But after the PstS16, I really like to walk in the field where the breeding trials are of our East African breeding pipeline, also the lines that are sent from Mexico as well. So some of those lines that have been like resistant for yellow rust, for stem rust, I select those lines. And then those lines I take actually forward to develop the biparental populations to see what kind of resistance they have, because some of the lines in these trials looked really, really promising last year when I was there, and they were like completely immune. So for stem rest, we can understand the presence of major genes, but for stripe rest, it looks really, really promising, some of those lines. So I've already made crosses, and they are actually in the process. So hopefully we'll get something good out of it. So for my GWAS study, what I'll also be looking at is like the candidate genes. This is a tool that I really wanted to show everyone that this is a tool called as Pretzel, which was also done by the Agriculture Victoria in Melbourne. And I think this gives you a really good comparative kind of like for the physical and the genetic distances that you have for your mapping. They have like put a lot of markers in there. You have 90K, you have 40K, you have targeted GBS, you have DART-seq markers, you have SSRs, you have STS. So anything that you are working on, you can actually put that here, and then they will tell you that where in the genome is actually that your gene or your QTL is present. And along with that, if you put a gene, I just gave an example of like, I've put like SR22. So some of these genes, they have already added into their tray, like the chip as well. So if you kind of like zoom in, it gives you the high confidence, low confidence candidate genes as well present in the region. So this makes the life very easy to kind of like take it forward from there and then see that what kind of genes are actually present in your region. So I think in the end, I would just like to say that like in the war against rust, we have to be really careful not to bring the susceptible varieties. So for me, I think that the gene discovery stays the most important thing that we can do to battle the wheat rust. And I think in the end, I would like to thank everyone. There are a lot of people that CIMMYT does collaborate. We do like collaborations. And I think I forgot to put JIC. I'm sorry, Diane. I kind of realized it when you were talking because I made it last night. So, but yeah, so I am sorry if I have missed anyone. It was not deliberate, but thank you so much for all the collaborations, for working together. And that's it from my side. Thank you so much.